really are waiting for the guys to give us a really awesome, cool intro. Anyway, welcome to Two Real Cryptid Legends with Darrell, Denton, and Ron Morehead. I am Michelle Desrochers, your moderator, host, and segment producer. We are broadcasting live on the United Public Radio Network, 105.3 FM from the Gulf Coast, 107.7 FM from beautiful New Orleans. Without further ado, let's get our hosts on board. Darrell and Ron, hello.
Hello, Michelle. Thank you.
Pleasure as always. Pleasure as always. Really looking forward to this show. I know you guys are really looking forward to this show because you have two incredible guests tonight. And do you want to do the intro? You want to do the intro? Tell everybody the surprise?
Yeah.
Go ahead. Yeah.
We have Dr. Melba Ketchum here, founder and the Genome Project, and then we have Robin Haynes, who is one of her closest friends, and both of them involved in the DNA studies of Sasquatch, Bigfoot, and we're so very blessed to have them both here tonight, and honored to have them.
Well, without further ado, shall we bring them on?
Absolutely.
All right, here we go. Robin, Dr. Melba Ketchum, welcome to Two Real Cryptid Legends.
Thank you so much. We certainly appreciate both y'all being here. Thank you, Dr. Melba Ketchum. Thank you, Robin Haynes. We really definitely appreciate you being here. We're looking forward to this for days now, so it's going to be a great show. Before I get started, Robin would like to say something. If you would, go ahead.
Thank you. Everybody, we really appreciate all the support that you guys are so great about giving. When the show is over, what normally happens when Dr. Ketchum does a show is that we get a lot of people that, while being kind and supportive, they message. And I just want to let everybody know, when you go to message Dr. Ketchum, you are not getting Dr. Ketchum. You are getting me. Okay. Right now, she is incredibly busy working on the Cryptid Genome Project. She does not have time to answer anybody's questions. Not that she doesn't care, not that she doesn't want to, but we need her focused on the work at hand. So she is not responding to anybody in her messenger. I am the one that is answering. So just an FYI, if anybody, is thinking about doing it, or they normally do afterwards, unfortunately, you're not going to get Dr. Ketchum. She is just far too busy right now. It is nothing personal. And again, we appreciate the support. The only person you're going to get is me. And depending on how many messages come in and we get flooded on any given day, is whether or not I can get back to you or how quickly I can. So I just wanted to kind of let everybody know your thoughts, your wishes, and the questions are appreciated. However, there's no way to get everybody taken care of.
All right. Thank you, Robin.
Yeah, thank you.
All right. If I could, I'll introduce Dr. Melba Ketchum. Melba, can you tell everybody how you got started in this scenario with Bigfoot and the DNA Project?
Oh, about a million times this has happened. I did not believe in Bigfoot. I thought it was silly. I thought maybe there might be a Yeti somewhere in the Himalayas, but I definitely just knew there was nothing in the North America that resembled anything like that. So I had a big laugh when our lab, we did a lot of different services and forensics in our lab. And every year we'd get a sample or two that was labeled Bigfoot. And I tried not to test them most of the time, but people insisted that they were, and they wanted them tested. So, one or two a year, and they'd always be something common, a goat, a cow, a horse, something. And we would laugh. We thought it was funny that people would actually send these samples in and insist they get tested. And that came up until 2008. And two things happened that triggered this. First of all, I was submitted two samples. That one was an eyewitness sample, The other sample had broken into a pen of peacocks and killed them, plucked them, and then took off with the bodies and tore through the chicken wire and left, huge footprints about, I forget, 20 inches long, huge footprints. And so I still didn't believe it. But we tested them. And about that time, we also had a sample from Destination Truth from Baton. And so we tested them all together and they all came out human. And I thought, no, this is not human hair. This looks like horse mane hair, about, anywhere between 3 and 6 inches long and wavy.
Were those samples from Texas?
No, they were from California.
Okay.
The first ones were. And so, in fact, David Pilates got them somewhere. I don't know who he got them from, but he actually sent those 2 in. But anyway, I got a little bit of an anomaly on the Destination Truth sample, but it still was primarily human. And they really kind of edited up what I said about it to make it sound a lot more exciting than it was. But the 2 Bigfoot samples were very exciting because they were obviously not human hair. So we retested them multiple times, trying to figure out how this happened. Of course, in a forensic lab, we always run a negative control and a positive control, as well as knowns from our laboratory personnel to make sure that, it's not contaminated. Plus, you can tell if it's contaminated because you would get the way this looks. Actually, I have a video on YouTube on contamination where I showed what it looks like and how I can prove it wasn't there. You have these like little peaks, up and down peaks, and if you have a contaminant, you're going to get superimposed peaks. You're going to see the little lines go up and down, and then there'll be one under it that's usually smaller. So a major contributor and a minor contributor, but you've got 2 samples in there. Well, these were clean, and I just couldn't figure it out. So I sent it over to my colleague and second author, Dr. Pat Vojkovic at the North Louisiana Crime Lab, who's one of the best in the world as far as these types of things. In fact, he wrote the book on the type of testing that was done to identify the tomb of the unknown soldier. So, and we use his techniques too, but he had better equipment because Louisiana was very, they were not frugal with him, and he had some state-of-the-art extraction techniques that were robotic that we didn't have. So we had 2 different types of extractions, and still we got weird human results with nothing else. So that's what lit the fire. And then when Destination Truth came on, we started getting literally hundreds of these samples from everybody because they found out that our lab would test it. And we were overwhelmed, and we managed to get some funding. I mean, I played with it for a while, and then when we got funding, I made a full project of it. And we went in there and screened all these samples. And we did it first by looking at the hair, had a hair expert, a forensic hair expert, do that. And they were unknowns. And they were, so we separated all the ones that were identified and kept the unknown ones to test. And there was over 100 samples. So that's how we got started. That's how the project got started. And curiosity, you know what curiosity did to the cat? Well, that's what happened to me because I got sucked into this thing because of my curiosity and no other reason.
But your credibility also went back, if I think I'm correct, with the state of Texas, what you did just criminal investigations with your DNA.
Oh, yeah. I've done them all over the place.
Right.
I mean, we've even had a British case. We did, all over the country. I, went to Hawaii and then all over the continental U.S. testifying in court. We did death penalty cases. We did cases that were Innocence Project cases. We did World Trade Center analysis. We had
Right.
a long history.
Yeah, I've been a follower of yours since the Genome Project started. Of course, late Scott Carpenter was a good friend of mine, and I followed you then, and I knew pretty much what was going on. I wasn't involved in it, but did help Scott with one of the hair samples. But that particular issue, when it all came down, I thought it was incredible evidence, and well, the credibility thing is what really got me upset, because your credibility is outstanding. I don't know how anybody could say you wouldn't be credible.
Well, I know, but they've done it. Believe me, the government set me up and did all kinds of terrible things. You'd think I was the devil incarnate if you look online between the government
I've heard
and
it's
the haters.
really terrible. So could you tell us what the DNA sample showed?
Well, it wasn't just it was
There was
bunches.
numerous. There was 100 and something, I believe, samples. And
Right. We didn't go through the entire testing on all of them because there were just too many and it would cost too
right,
much. But we took about 30 of them and proceeded through that group, and all of them were consistent with human mitochondrial DNA. That was the first finding, which is the mother's side. And depending on the sequence you get when you sequence, and sequence is just the order that your DNA has of the 4 nucleic acids that make up DNA. And that, and they're just abbreviated down. Thymines a T and guanine's a G and cytosine's a T and adenene's an A. And the order of those letters on your printout that you get from your sequencer determines your genetic makeup. And even one base different can make you a different individual. And and they're called point mutations, or single nucleotide polymorphisms, SNPs for short, SNPs. And that's primarily what we use on the mitochondrial DNA, is that you can make a tree starting with the mitochondrial Eve, which has one type of mitochondrial sequence. And then as years passed, a mutation formed in some of the offspring, and you can trace it from there where they come from as the mutations apply or form. And so you can see where the original ancestors came from. And that's how we know that most of these samples, the majority of them, were either Middle Eastern or European, because Middle Eastern branched into Europe. Now they branched the other direction into Asia, and that's where the Native American mitochondrial DNA comes from. But we only had a couple of those, and there's a caveat there, because we don't know when that hybridization occurred. It could have occurred in the last, 500 years.
right.
It could be we just know that the mother that's showing up is whatever it is. But the vast majority of the samples were from Middle Eastern origin, easing into European origin. And most of them were 13,000 to 16,000 years ago is when they started. Those particular groups started. So that's how we're able to tell how old the origins are. It's also how we can tell where they're from originally, the mothers at least. Do you understand that?
So I understand some of it, not all of it. I wish I understood. I'm sure there's some in the audience that don't either. But can you tell us what the microchondrial DNA is? I mean,
yes, we have more than one type of DNA in our bodies, and on a cellular level, you have, like, if you can imagine what a cell looks like, it's a round. Well, not all of them are round, but basically it's a round organism with a nucleus, which is a kind of a circle shaped. It's like a ball inside it. It's in the center of the cell. It's what contains your chromosomes and your DNA. Now, outside of that is what's called the cytoplasm, which is basically the liquid surrounding the nucleus, and it's got little things called organelles in them. Now, organelles are like little organs, and they do different housekeeping things for the cell. And one of those organelles is called a mitochondria. And a mitochondria produces energy for the cell. And it has its own little small loop of DNA in it. It's called the mitochondrial genome. It's not the nuclear genome that has so much of the genetic information. It basically just has the loop of DNA that interacts so that you can get the energy for the cell to survive. And it's only from the mother. And there's a reason for that, but it's too long to go into. But
yeah, I understand. Yeah,
but anyway, there's a 16,000 base. There's like 3 billion base pairs in the nuclear DNA, and there's 16,000 in the mitochondrial DNA, thereabouts. So, but it is from the mother. So that sequence there will trace back to the maternal origins of any creature.
So what was the final outcome of the Genome Project that you come up with? What formula?
On the mitochondrial DNA?
Yeah, I mean, is it human female, or I think that
It's 100% human female that first came into existence 13 to 16,000 years ago. I mean, we had one outlier here and one outlier there, but you have to kind of rule those out because we don't know when the hybridization actually occurred on those. But you can look at the one that's most of it and pretty well hang your hat on that's where they came from. And so that's what we did. But they were 100% human female of Middle Eastern origin. And the later ones were European origin, which is one of the migration paths of the Middle Eastern DNA. So it all lined up. We all expected to see the Native American groups in there, but they just weren't there. I mean, there was one or two, but they just weren't there. So those were outliers.
I guess there could be a possibility of some of those not being there at this particular time. Could they have been there at another time and not showed up in the current DNA?
Well, I don't think so. I think we're seeing where they came from. However, once again, I'll put this scenario forward. Say that a regular Bigfoot, and this is in legend, especially in the Pacific Northwest, that they mated with Native American females and had children at times. And say that this was one of those examples from, the Native Americans have been here for 13,000 years and longer in some cases. And so anytime during that time, that mating could have occurred. And if she had the Bigfoot's baby, then it's going to show her mitochondrial DNA. So the fact that it is so few in number compared to the number of samples, though, makes it so that it's not going to be the origin. Same with, we had like one black one, I think, and it came from the southern U.S., and we actually think that most likely this was a slave that had a child, probably ran away and got taken in by a clan of Bigfoots and ended up with a child. And that's how we got that one, because we only had one of those out of all of these. So, it's like you have to do statistically what's best. And since almost all of them were the Middle Eastern going into Europe, those pretty much have to be the origin, if that makes sense.
Yeah, I think I understand. I hope the audience does too. So what would your percentages be on roughly, so how much percentage would be human female and so on?
Well, it's 100% human female. It just depends on where they came from.
Okay. All right.
And every single sample we tested was non-human hair yielding human mitochondrial DNA results.
Okay. And
Was it primate hair?
I'm sorry?
Was it primate hair?
Well,
Was the hair considered
not,
primate?
no. It's unknown.
yeah, so the father was unknown species.
Yes.
Okay. Yeah.
And the hair is something that has not been seen. And everybody says, oh, it doesn't have a medulla. They most certainly do.
That's what I was going to ask right there. Yeah.
But the problem is, and this is something unique that I've never come across, and I haven't found anybody that's ever come across it, unless it was a real teeny tiny fine hair like a newborn baby or something. These hairs have medullas, and you can get mitochondrial DNA out of hair shafts. You don't have to have the root. But we spent 3 months because we couldn't get any mitochondrial DNA out of the hair shafts, big fat hair shafts with medullas, and we couldn't get any DNA out of it. We couldn't figure it out. I mean, I tore everything apart. I replaced all the chemicals. I did everything I needed to do. I sent it to Dr. Vojkovic, and he used his robotic extraction methods. He couldn't get any DNA either. We finally put it on a spectrophotometer to see if it was an inhibitor keeping the reaction from happening on the hair, or if it was no DNA there, and there was no DNA there.
Wow.
And nobody can explain that. In fact, the psych study that set out to disprove mine, they had 7 samples that didn't run. And in the paper, he says, we don't know why they didn't run. He did know, because he had read my paper, and we addressed it in the paper, that the hair shafts will not give DNA for some unknown reason. And he, admitted they didn't know why. And the thing was, most, if not all, those samples were samples from my study that the owners had submitted to them just to see what they'd get. So all it did was support our paper.
Melba, out of the 100 or so that you looked into, how many of those were from the West Coast and how much were not?
Well, they were from all over. I mean, I had them from so many different states. If there was woods there, I had them.
No, I know you had some from Tennessee. I know you had some from Kentucky.
Yeah, Alabama, Louisiana, Texas,
Michigan.
Oklahoma. Yeah, Michigan. I mean, you name it, we had it. If there were, I mean, if there were trees, we all along the West Coast up into Canada and Alaska. I mean, we had a little bit of everything.
It's pretty incredible.
Well, there's a lot of them. People don't realize there's a lot of these things out there.
I know they are.
Everywhere.
I have no doubt about that. And I know that you're very aware of that. I know Robin has a client she stays in touch with, and I know you do too, Miss Melba. But these creatures, I got tickled. I was at a Bigfoot conference with Ron back in, I don't know what year the Florida conference was we were at. Ron, what was that, 2022? Maybe, I don't know, but
I don't know. I don't remember.
Anyway, I heard a well-known speaker say that there might be 4,000 of these creatures in the U.S., and I literally started laughing because
I know, you can't help it.
I think there might be that many in Middle Tennessee, but the numbers are incredible.
People come up to me all the time, and they're like, I figure there's only a couple thousand of them across the United States, and I'm like, try like within a couple counties. Like, people have no clue. It's like, be at my house. Okay, they're everywhere, but they're just everywhere. There's no place that there aren't. I have clients that are actually in Hawaii, and there's a smaller version there, but they're there. I mean, they're everywhere.
And they're not limited.
Yeah, they're not limited anywhere.
And they're all across the world too, just in a different type. What is it? A rain pendak, I believe, is in the Asia area, and it's just a smaller version of it.
That's another
Australia.
thing. I'm sorry, go ahead.
No, go ahead. That's fine, Melba.
I was going to say that Dr. Tom Gilbert out of Copenhagen actually tested an orang pendek sample, and it was bright red, of course.
Right.
And it came from an area where there's no red-headed people because they're dark-headed natives. And it came up human also. So it supported
Wow.
what I'd already done.
Wow.
Really? Yeah.
So
That's
there,
amazing.
I had a Russian sample also that turned up human, but they wouldn't, it was out of destination truth and they wouldn't let me test it for this.
Well, I know you and Robin have went to Russia and did some studies over there. And
She did. I actually went to adopt 2 children, so I didn't go for the same reason.
oh,
I
okay.
was over there with Robin.
Yep, Ron was there with me. That's how I met Ron. We had fun. It was great over there.
That was.
Still talk to those ones in the cave. They still will mind speak. And they
Mm.
always start out with Russian. And I'm like, people have tried to teach me Russian for years. I cannot do it. I cannot. Just English. And they'll either go between their own language and mine. But yeah, they still stay in contact over there.
When we
That's
went in
amazing.
Siberia, remember the shore people over there? SHOR? They
Yes.
sent us to Mongolia. Yeah. Took us into some really interesting spot, way back in the middle of nowhere, across
Yeah.
rivers and everything else. That was fun.
That was.
Very
It was amazing.
We found that track in the cave. And anyway, I just wonder if the genome would be the same for those in other countries like that, if they would all be somehow related. Do you have any opinion on that, Melba? If one's outside the area that you've checked the genome on the ones, DNA on the ones that you were sent, but do you have you ever heard of the one out of the UK where the geneticist at above the Bhutan, I think it was, at 17,000 feet, got some eDNA, environmental DNA, and checked it, and she came back and said it was human. And that was 17,000 feet on a bipedal track in the snow up there, or ice, I should say. Have you heard of that one?
I'm sorry, I'm having trouble understanding. I'm getting a lot of rubber banding.
Oh, I'll pull my rubber bands in. No, there was
Audio is bad. Let's put it that way.
a
The
National
audio is bad at
Geographic
the moment.
study. A National Geographic study went on several years ago up above Bhutan at 17,000 feet, and they had a geneticist, a lady up there who got some eDNA out of some tracks that they found. That was at 17,000 feet, and she came back a couple years later and announced that it was a human. The mitochondria was human, and that
Well,
was really. I just wonder if you've ever heard of her or knew about that.
no, I actually didn't know about that, but I don't pay a lot of attention to other people's results necessarily with it, because I'm not a believer in using that type of DNA for trying to prove anything, because there's too much a chance of contamination. And if she took it from the snow or soil or whatever, there's no way it's going to be a clean sample. That's why the second study I'm doing, I'm only using hair samples. And the reason for that is you can wash a hair sample and get any contamination off. We have high-speed vortexers. We have all these different types of equipment that will guarantee shake any cell, any DNA, any tiniest of piece of DNA will come off of it. So you have an absolutely pure sample with hair. You can tell a lot about the samples that are the environmental DNA, but and I've dealt with them and I've gotten good results, but it's nothing I feel that you can take and prove it with. And the reason I say that is because everybody screams, oh, it's contaminated, it's contaminated. Well, at the very least, you're going to have all kinds of sequences from the dirt, all the bacteria in the dirt. You're going to have molds, you're going to have viruses, you're going to have all these things. Now, if you get a really good sample that's high in DNA, like saliva, we use saliva, among other things. You can have such an overwhelming amount of the DNA that you're looking at that you can pretty well prove it. But most of these environmental samples are not that high laced. They're going to be just a few cells, and so you can pick something out that doesn't belong there, yes, and that can work to an extent. But you're going to have every critic in the world wanting your head on a platter, saying, "Oh, this is just garbage because it's got all these contaminants." And you have to know what you're doing to deal with it, first of all. And if you know what you're doing, to prove anything at all, you have to use something that's so like I'll use our saliva samples. And I've never talked about this before, but I'll say this, that the saliva had so much DNA in it that we got a really good profile of the Bigfoot when we did the whole genome. But yet it's so down at less than 1%, you could actually see the peanut butter DNA,
Wow.
the bread DNA,
Wow. That's
some
amazing.
spinach DNA, and you could actually see also some deer DNA. And these were so little that they were statistically unimportant, but I thought it was great because we learned what it was eating. We knew it ate the peanut butter sandwich to get the saliva. And it was fixed in a way where it had to lick the plate.
I believe there were some blood samples, weren't there?
Samples, and once again, they were high enough that we were able to very much use them and differentiate. But in an average where you just find a footprint, you're not going to have the amount of DNA necessary to do a good job
Right.
of proving anything. It has to be like, 99% of it or 95% of it at least is going to be the target in order to prove anything. So, I'm happy with our results on it, but as far as I would never try to take one out of a footprint unless it was a hair, in order to try to do the environmental DNA, because it's just not going to have enough DNA from the target animal to do a good job. That's all I'm saying. Because we got some flack from people over that on some of ours. But I can guarantee you there was enough of the target Bigfoot there that we had really, really, really good results and consistent results. So, which I haven't even released yet. They are coming, though, I guarantee you.
Interesting.
So, I don't keep up with anything that's environmental, unless it would be something that they found, like it cut itself and there was a bunch of blood or something.
Well, we didn't use the blood sample from the one that got cut at my house, remember? Because he got cut. It was one of my group, and he was walking back to the woods, and there was all that blood in the snow, remember? Because Igor grabbed it, and we were going to use that, and there was just too much contamination. It wasn't worth it.
Right. That's why we didn't do it, too.
Yeah.
Like I say, the only ones that I used were, like I say, those saliva samples. Saliva's high in DNA. And so it's a very good sample. I don't want to use it this time because I'm looking for some specific things. But as far as for the original one, yeah, it worked fine. And we had a lot of DNA there so we could go further with it. So, yeah, I mean, as long as you have something and you have something, like we used a sample that was blood, that had cut, but it was cut on and it flowed into the inside of a drain pipe. And by doing that, it wasn't like people may have handled the outside of the drain pipe, the gutter, but they wouldn't have handled the inside of it. So we were able to slice that open and get a clean sample there too. So,
wow, it's amazing.
It depends on the circumstances, I guess is what I'm saying. Because you can't wash saliva and you can't wash blood. So you have to take any contaminant with it. But the other good thing about both of those, that there was absolutely no other mammalian contamination other than the teeny bit of deer that was so tiny that it was once again statistically not important.
Wow, it's amazing. What you say you are currently working on a new study? Am I understand that correctly?
Oh yes, we're doing more than just the Sasquatch though. We're doing Dogman. I've got some really good Dogman samples. Some of them, one is completed, one is almost completed, and the third one's it's slowing things down a little bit because I got it late, but it's from an eyewitness. It ran up to a little girl and kneeled down by her, and she had her dog with her, and the dog bit the thing and pulled a bunch of hair out.
Wow.
And so I'm looking forward to seeing.
Can you tell us what state it was in?
I'm not going to say right now, but you know, it was an area where they have them, and obviously, well, they're everywhere, but it was a more wooded area. I want to say, let's just say the Illinois, Ohio area, and leave it at that. And there's a lot of cryptids up there.
Yeah.
But
so many cryptids. Oh,
huh?
I said there's so many cryptids up by the Ohio.
Yeah.
I get calls all the time. I wish we could get samples from all of them.
Yeah, this is something I'm still looking for samples on, anything that's unusual. I've got a couple of giant samples. I would like to get more. I would especially like to get one off of a 6-finger, 6-toed one with a double row of teeth. I would love a tooth from one of those.
Wow.
I would love to have a mermaid sample.
That's the gugway there, the face eater.
I got a track from 6-toed Bigfoot.
I'm sorry?
I don't think it has a hair in it though. I got a cast from a 6-toed footprint.
Oh wow!
From a Native American up close to where our camp was. I mean, within a few miles of where
you can look for hair, but it's got to have a root on it. It can't be without a root, or it won't run. That's the issue. Other thing, Goat Man. My favorite that I'd like to do is a mermaid.
I'd love to do that one.
I would love to do that. I wish somebody would have one, because a sample.
Let's go to Hawaii. I think I know where they're at over there.
Well, go get me
a sample, Ron.
Yeah, just dive right in.
Get over there and get something.
Still right where they were.
Well, I would like to know if their other parts are fish, or are they mammals, like a dolphin. I would like to know what their makeup is.
Yeah, that'd be interesting. How about an octopus?
Somebody offered me that, but I couldn't confirm any information that made me feel well enough about testing something like that. If you know more about it, I'd love to hear it.
I think I got 8 brains, 1 in each tentacle. So. But,
I'm sorry?
Are
you talking about one in the ocean, or are you talking about a land octopus?
I'm talking about one in the ocean.
Oh, okay. All right. I was offered one that allegedly was a land octopus, and I just couldn't. I researched it, couldn't find any information, and I was like, I don't think so. Because I have to, of course, watch the research funds. I don't want to just throw them away after a bad sample.
Yeah.
So,
when do you expect your project right now to be out? How long do you think it might take?
I really don't know, because it's a start and stop thing, and I got to be so careful about sending them out so the government doesn't snag them. I had one good sample out of Australia that got snagged, and so I've got to be very careful about all of this. And so, I'll get some stuff done, and then it'll slow down, and then I have to analyze what's done and put it together. And that's why I really haven't given a lot of updates, because I've got different samples at different stages, and I don't want to prematurely give away something that is going to possibly change. But, say I've got one sample showing something else, and then I get one that shows something different. I'm going to have to figure out why it's different and if it's real or not. So, I mean, there's just a lot of work to it. And plus, then you got to have the bioinformatics, of course, which is what we had done on the original genomes. And it took a couple of years to get all that done. But when it was done, it was fantastic. And I've got beautiful pie charts of, what was in the samples.
Wow.
So, but once again, the guy that did them caught flack. So I've got, I'm having to use someone else. And so that's not good. The other thing I have to do is I generally change my name when I submit stuff.
Yeah, I can understand that. Could you tell the viewers out there, the people watching, that is there a credible place that someone could send their DNA to?
Well, for the first screening and the first testing, I would definitely go with Paleo Labs in Ontario.
Okay.
They're a good group and they're not prejudiced against these type of samples. So they're a good lab.
I get asked that at least once a week. Somebody will message me, or they'll have some type of sample. And I know you and I had talked about that a week or so ago. And some DNA samples you would suggest not even seeing, like scat and that type of stuff.
Scat, unless you catch it when it's smoking and you immediately put it in a baggie and freeze it, you're not going to get anything that's helpful.
Right.
Plus, there's no way it's not contaminated.
Right.
You're more likely to get, say it ate a deer, you're more likely to get the deer DNA than you are the Sasquatch DNA, because, and if you do, the Sasquatch will be on the outside of the scat. It won't be inside. Inside will be what they ate.
Right.
Pretty much. So you want the outside, but I just think it's a waste of money, to be absolutely honest, to do any of the stuff.
I agree with what you tell me about that. So you would suggest the best DNA would be either hair, saliva, but one of those two would be
or
the
blood. But the thing is, you have to make sure that, like with Robin's sample, it was in snow. And you could have taken the blood from the snow and tested it, but it got handled. So there was no way to wash it. So we knew it would be contaminated before we ever started. So there was no point. But like these, the one that had that ate the peanut butter sandwich, that was handled with gloves and sterilely, because that's what it was set out there for. So there wasn't. And when we did the sequencing on it, I looked for any type of visible, this was the early part, whenever you do the original sequencing, the first screening of the mitochondrial DNA. I looked to see if there was any type of human underlying mixture there, and there wasn't. It was perfectly clean. And the deer would not have aligned with it anyway. It would have put up a whole bunch of different peaks, and if there had even been enough, but there wasn't enough deer in it to show up on the sequencing, the original
Melba,
sequencing.
Can you tell everybody, because I get this question a lot, how do they store these samples? Because like with the blood that was taken at mine, I was not the one that gathered the sample. It happened at my house. It was one of the group that I work with. I was not the one that collected the sample. It was collected wrong, and we were not able to use it, which was fine, not a problem. But a lot of samples, I get people that contact me all the time, and they want to know, how do we store them? What do we do? Another big question I get all the time is, I've got all these stool samples, and that's not the best sample. But I was just wondering if you could take a minute, kind of maybe tell people,
Go ahead.
No, just kind of give them an idea.
Well, it's very simple, actually. Always have your gloves with you in case you come across something. And you don't want to stick them in your pocket or anything. It's best if you have surgical gloves. But if you don't, just get you a box of gloves from Walmart, and be careful that you don't handle the hands on the gloves when you put them on. Put them on from the cuff so that you're not rubbing human DNA all over the gloves. And once you got your gloves on, you either flame you got tweezers you're going to pick it up with, then make sure they've been run through a flame before you do it. That will kill any DNA that's on them. So you can take your tweezers, you pick up your hair, have a brand new paper envelope ready and put it in that, because that'll let it keep it dry, and dry will preserve DNA for forever nearly. So that's the way to preserve it, is just to, if it's hair, put it in an envelope. If it's something wet and you need to put it in a plastic bag like saliva or blood, then same principle as far as doing it cleanly, and brand new Ziploc bag that you've not touched the interior of at all. And you put it in there and you get it in a freezer or an ice chest as quick as you can. And do not let it stay out. You don't even put hair in plastic bags, because plastic bags hold moisture. And if it's moist out when you take the sample, it can absolutely have enough moisture in it, especially with temperature changes, that'll condense in there, and then you will ruin your sample, because wet will breed bacteria which will eat your DNA. So it's always
Wow, it's
better
great
to have
information.
a dry sample or a frozen sample. And other than that, no.
I have a question. If a person, if a sample gets contaminated by an individual, wouldn't that individual's DNA be on that sample also?
Yeah, of course it would. That's why I'm saying that hair is the best, because you can wash the hair and get any contaminants off of it. If you picked hair up with your bare hands, I could make a clean sample out of it just by using my vortexer and various chemicals to clean it.
Mm-hmm.
But the root on the hair is not going to go anywhere because it's attached. Whereas contaminating DNA is loose.
The people that claim some of these specimens are contaminated, wouldn't their DNA be on that contamination? I mean, we could have shown that, right?
I did a whole video on it on my YouTube channel about contamination, because once again, you've got these little peaks that the machine puts out, and they go up and down, and they're pretty close together, and they're color coded for each nucleic acid base. And of course, no two individuals, well, you can't, like a mother-daughter or a mother-son, they're going to have the same mitochondrial DNA because it's passed down generationally from the mother. And so they're going to have the same one. So if a mother contaminated a daughter's sample, then you won't see any mixture. But if, say, you contaminated a sample that was a Sasquatch sample, both have human mitochondrial DNA, but yours is going to superimpose with the sequence of the Sasquatch, but you're going to be different. Say you have Native American lineage, let's just say for purposes, you're going to have different point mutations. You're going to have, like, you'll come along and you'll see a peak that's a T and an A and a G, and then you come up with a C, but about halfway as tall as that C is an A, and that's not supposed to be there. It's very rare for there to be an accidental, and that's called heteroplasmy, but that's accidental. But it's thousands of bases before you come across heteroplasmy. Whereas you're going to see, 10 or 20 bases that could be different. They'll have these superimposed peaks, and that clearly shows contamination. Clearly shows a second human being. And there's no way around it. That's how you do it in forensics. You're going to have the suspect cuts himself when he stabs the victim, and the blood mixes a little bit, but it's mostly the victim's blood. So we run the sample. In fact, we've convicted people using this, where you've got a nice clean sample that matches the victim's known blood, and then you have a known from the suspect, and the suspect will have, say, a peak that's about 2/3 as tall as the peak that belongs to the victim. And you can match the sequences that way and actually determine that the blood is a mix of, and it's contaminated, therefore, it's a mix of the victim's blood and the suspect's blood. And you can convict people that way. It's quite easy, actually.
Wow.
And so I know when there's contamination, I know what to look for. I have the electropherograms, which is the name of the graft. I have those to prove it wasn't contaminated by any other mammal or human per se. And so when it's clean like that, that shoots their theory down, but nobody would let me show that. I offered it. They're like, oh, we don't need that. Yeah, you do, because everybody says it's contaminated, and it's not. In fact, in my video, I had to use another sample from a crime scene, or I think it was from crime, either that or I made it. I can't remember. But I made sure I had a mixture and ran it to compare it so that people could see what contamination looked like versus the clean sample. And I used an actual from the study for the clean sample, but I didn't have a contaminated one from the study. So I had to make one or use one that I'd already tested. I can't remember which, to show the comparison. So anybody that has a question about contamination, go watch the video.
How can we see that, Melba?
I mean, I've done a few things like that. I'll always defend the paper. I know the science we did.
Is that available on your YouTube channel? That's what Ron was asking.
Yeah, it's at Dr. Melba Ketchum.
Yeah.
At Dr. Melba Ketchum. Okay. Thank you.
I've got, several goodies on that. I hadn't done anything recently on it, but I had done several goodies. But
you mentioned your new study that you're doing, Melba. What are you trying to determine on the new study?
Well, actually, and it's already sort of showing that direction. I'll give this much out. I wanted to show relatedness against cryptids, and I believe that most cryptids have human mitochondrial DNA. We've already confirmed that clearly with dogmen.
Wow.
So, that their mitochondrial DNA is human female. And there's no doubt about it, because that's repeated.
Let me hear that again. The dog man's human, the DNA.
The mitochondrial DNA, the maternal mitochondrial DNA is human, just like the Sasquatch.
And for anybody that mind speak, ask them. They'll tell you.
Right.
That's how I met Melba, because I didn't know her before the study. I found out she even existed, and the study existed because the Sasquatch told me to find her. They didn't give me her name. They just said the woman with the blonde hair knows the truth. Find her and help her, which was ridiculous because I don't know science. So, but, I helped her any way I could. But if you talk to the dogmen, if you are mind speaking to these beings and you ask them, they will tell you. They're not shy about it. And she's gotten it right from every single angle that they said.
I've known Robin for quite a while. She's very incredible with her mind speed. So I definitely would believe that. But that's pretty amazing. I've kind of heard people mention that in the past, but to know that it has the same as the Sasquatch with human female, that's pretty amazing.
I can tell you this: the nuclear DNA is going to be different than the Sasquatch. Right. I can tell you that much. But the mitochondrial DNA.
What alien created them?
Yeah. Oh, we wish we knew.
Yeah, and I really appreciate all the help Robin gives me, because she saves me a lot of time so I can do things I need to be doing.
Yeah.
And
we make a good
she does
team.
a great job. Huh?
I said, we make a great team. You do all the science and I'll do anything else but the science, because I don't know it.
She does a lot of the administrative stuff for me that I have no desire to do, to be honest. And, like she would send out things for me and emails and does a lot of correspondence for me. And I appreciate all that, because I just really don't have time to do it. And I'm not bent that way. I'm a scientist, not somebody who is very great at wanting to do the administrative end of things.
Well, I
Right.
think the Sasquatch are really good at picking out what they want done because, like I said, I didn't know she existed. I didn't know the study existed. They told me to find her. I had no idea what they were talking about. I knew somebody that had heard about the Sasquatch Genome Project and knew her by name, and I guess had exchanged messages or something with her at one point. And I was like, I don't want to bother this person. She'll probably think I'm nuts. But the Sasquatch told me to find her. And so they gave her my number and she contacted me. But we just work very, very well together as far as that goes. We have a lot of
Yeah,
fun, but we
and
work.
I very much appreciate her.
Yeah,
Well,
well, as I do you, sweetie. But,
Yeah,
Now,
you know, I will say this. I have had, and, you know, I don't know what's going to come up on the dad's side. I know the part about the mom, but they do interbreed. I have a picture of a female that is part
Sasquatch,
dogman. Yes.
Sasquatch and Dogman interbreed.
Yeah. Yeah.
Wow.
And she stayed with the female Sasquatch and the male dogman. The three of them stayed together. And she hung out at my property in Michigan. I have a picture of her. And she looks like a
Wow.
teddy bear. She's got the teddy bear face. And then she's got the Sasquatch body, but she had a very, very long tail. So at some point, I don't know what it's going to come back at, but at some point they're able to breed.
Wow.
I
I
mean,
didn't know what
the
it,
chromosomes has to match up. Yeah. Wow.
So there's so much weirdness about this. I'm going to give one more little thing for people to look forward to.
Melba,
Um,
will you be checking the nuclear DNA on these samples that you have, the dog man?
oh yeah,
Uh-huh.
absolutely. I mean, I
That's
already
pretty
know
good.
what it is. I just want to wait on that third one to make sure
Uh-huh.
it lines up with the others.
wow, that's
That's
amazing.
fantastic. Yeah.
That's amazing.
So,
It won't be in the gene bank, will it?
I'm sorry.
It probably won't be in the gene bank, will it?
They didn't let us put it in last time. See, that was another criticism. Oh, it's not in GenBank.
Yeah.
I have the correspondence where they wanted signed, since it came back human, they wanted signed releases from the sample givers. They wouldn't let us put it in.
Hmm.
I mean, they gave us every runaround in the book. My third author handled that, and she was frustrated beyond all conceivable belief.
Pretty amazing. You got somebody that the state of Texas has enough confidence in with a DNA study to put somebody in criminal position to go to prison for the rest of their life, and they don't trust you with your DNA samples.
But see, that's the whole thing. Also,
so
is that
ridiculous.
it wasn't just me. I had a fantastic team. Dr. Vojkovic
Right.
is one of the best in the world. Like I say, he wrote the how-to on the reassembling the mitochondrial DNA to get tiny fragments together so you could identify very old remains, like the tomb of the unknown soldier that had been out in the jungles for forever. He did the one from Vietnam, and it had been out in the jungles and was all degraded. But you can use that particular procedure and get it done. We used it in one of our criminal cases.
Wow.
That was it was a dog that had been buried for 15 months. A classmate of mine dug it up and he was cussing me the whole time because it smells bad. but we took samples off of that thing and it was so degraded that we ended up using Dr. Vojkovic's methods to get a good clean sequence, and it matched the suspect. So, the suspect, when they found that out, he confessed. And that was my favorite case of all because we worked the hardest on it.
Wow.
But, besides that, the thing is, you just can't. I had somebody that I was working with. They make people disappear. The government will threaten them. And one of them was told they can't spin DNA, so they have to do it like this, by threatening people around me and by discrediting me. And so that's how that goes.
That's too bad. But anything the government can't explain, they got to discredit it or.
That's exactly right. Yeah.
Well, it goes a lot deeper than that. I mean, if you think about it, I mean, how many people would want to go to national parks if they knew some giant hairy person was staring at them all the
That's
time?
always been my
Well,
theory.
that's
You know, the
problem.
national
Yeah.
park industry is a 2.8 billion with a B industry. So
Right. And not only that, you've got the more nefarious types, the fact that these creatures have abilities that Robin's well aware of, and I am too. That
And I am too.
yeah, I
Yep.
mean, everybody knows
Ron
they
knows
do.
it.
For the most part.
yeah,
You know, anybody who's been around them very much that they like, they're going to know this. And there's a lot of nefarious things about those powers that they want to know about too. So, plus it's hard to explain. It's like ETs. You know, how do you explain an ET to most people? You know,
I think it's all going to come out shortly.
oh, it's
probably
going
next year.
to. That's for sure.
Yeah.
I
The
mean,
disclosures are starting.
All those attributes of these things are showing to us, like they do with you, Robin, I have with me and Darrell.
yeah.
That's all within us also, because we are human. But we've been limited by our own. We haven't been
It's all
short.
the frequency.
It's all about frequency, and as soon as we can get our
Absolutely. Frequency and energy.
yeah, you can change your vibration, and that's what it.
And as soon as you raise your frequency and your energy to a high enough level, then we all can tap into the abilities that we have. It's not that hard. I teach people that every single day. And the cryptids just did it first. We were taught, because of the powers that be in our third dimensional world, that none of this exists. But the thing of it is the planet is shifting. The earth is shifting. We're going into that higher frequency and that higher vibration and the higher energy. And everybody's abilities are popping right now. They just don't know what to do with it.
Yeah. More than ever.
Yep. But we all can do it.
Yeah. It's happening.
Something I found.
We are at, we're past the top of the hour. Is there anything? I mean, there's a lot of comments and such. We can take a question or two if you'd like, or if you want to just tell us.
Thank Melba personally for coming on and talking with us. That's
sure, 100%. Her and Robin. I want to thank Robin and Melba.
Robin, you too. It's good to see you again, Robin.
I know, it's good to see you. I haven't talked to you in a while. I'll have to get hold of you and bug you.
Look at Nebraska, maybe.
I would love it.
I mean,
you need to come back. Are you going to come with Darrell?
I don't know. No.
Yes.
I don't like to travel like I used to. I mean, the commercial traveling back and forth just all
I know it's hard on you. I know it got hard.
This is it's hard.
Well, I've traveled roads with him many times. He's welcome to come with me to the Nebraska conference anytime. I'm honored to be invited back again this year. Thank you, Rob.
Oh yeah, we can't wait. We're excited.
I'm honored to be there, but I just want to tell you, Robin and Melba, thank you so much for coming on. It's really been awesome. I've learned a lot that I didn't know, and I'd like to hear more. I'd like to get you on again sometime when you come up with the evidence on your latest study. I'd like to have you both back on again. Ron and I would, and our great partner and moderator Michelle Desrochers over here. She's the best. So we appreciate it. If you want to answer a couple of those questions, you're welcome to before we go. If you don't, that's fine too.
Yeah. Otherwise, I know that Robin wanted to reiterate her message also.
Sure. Exactly.
Yeah. Again, you guys, and this is not, I want everybody to know we appreciate the support. We appreciate the love and the fact that everybody wants to talk to Dr. Ketchum. And trust me, from somebody that knows her, she's flipping awesome. But she is very, very hard at work on this next project, and she is not the one answering her messages on Facebook. So when you submit a question or something to her, please know at no time are you going to be reaching Dr. Ketchum. You're reaching me. And depending on the volume that comes in, is whether or not I can get to everybody. But you're not getting Dr. Ketchum. As much as we would love for her to be able to have that kind of time, she simply doesn't. And we need her talents used for the project. So the only one you're going to reach if you message her is me, unfortunately. And I'm sorry to disappoint everybody, but just wanted to let everybody know. And again, depending on how many messages I can take care of in a day is whether or not I can get to everybody.
Thanks, Robin.
Thank you,
Thank
Robin.
you.
Yeah, I thank you, Robin, too. And that's true. I really am, the first time ever, so busy that I don't have time to breathe hardly.
No, she does not.
Well, keep breathing. It's very important.
Yeah. You need to breathe to do this incredible work that you're doing. Well, we thank you both for joining us this evening, you guys. If I could just sit here and all listen to the four of you, it was wonderful. And to everybody in the chat room, thank you all so very much for your input, your comments, and your questions. You had a lot going on amongst yourselves, which is wonderful. So thank you. You have all been listening to Two Real Cryptid Legends with Darrell Denton, Ron Morehead, myself, Michelle Desrochers, with our two very special guests, Dr. Melba Ketchum and Robin Haynes. Thank you. Thank you. And you have been listening to us on the United Public Radio Network 105.3 FM from the Gulf Coast, 107.7 FM from beautiful New Orleans. We thank you all. Please watch the social media for the next show. Thank you. Have a wonderful week.
Thank
Good
you,
night,
everyone, for
everyone.
watching.
Thanks,
and
everybody.
Thank you, Melba. Thank you, Melba and Robin.
Oh, you're
for being
very
on.
welcome. Thank y'all for having me.
Oh,
And thank you, Robin, for helping me.
Always, I got your back. She covers my back, I cover her, so it works out really well. I don't know what I'd do without her.
And we don't know what we would do without you guys. So thank you all. And
Well,
good
thank you
night,
so
everyone.
much.
Thank you.